Alright, hi, my name is Dara, and today on Ethically Speaking, I will be talking about CRISPR and the future of gene editing. What can it do? What should it do? And where should we draw the line? So, what is CRISPR and how does it work? CRISPR is short for clustered, regularly interspaced, short palindromic repeats, and it is one of the fastest and least expensive ways to edit genes. CRISPR actually evolved in bacteria. So, when certain viruses infect bacteria by injecting their DNA, the bacteria catches the virus's DNA and it chops up the virus's DNA before it can cause any harm. Some bacteria even keep a small piece of that virus's DNA as a sort of memory, which is the mugshot. And that way, if the same virus ever tries to invade again, the bacteria recognizes it immediately and it launches a quick attack. So it uses in human beings and in bacteria, CRISPR uses what is called a guide RNA to find the target. And then this guide RNA attaches to the Cas9 protein. The Cas9 is the one that acts like the scissors, and it leads Cas9 to the exact spot in the DNA where it needs to cut. And after that cut, the DNA tries to repair itself by either reconnecting the ends or it uses the other strand of DNA as a template to fix the strand that has been cut out. And this really does matter because this is one of the best, cheapest, and fastest ways we have been able to edit genes in modern science history basically. So there are two types of gene editing. One is germline and the other one is somatic. In terms of germline, we alter DNA in the embryo and it is done in reproductive cells like the sperm and the egg. This kind of editing can be passed on and it is often done before the person is born, and the changes that is made in that person's in the embryo, it can be passed on to the person's future generation. And that is what is being showed on the left here. Germline edits, it affects the entire person's, or the entire person is carrying the entire edit. It affects their future generations, the kids that they give birth to, their gene pool, the kids that their kids give birth to. Meanwhile, in somatic, it only affects a small part of the individual. So it is used in non reproductive cells like skin cells, liver cells, and it cannot be passed on to their future generation. It only treats existing diseases after birth. And as is shown here in the picture on the right, this kind of edit doesn't work on the gametes like the eggs and the sperm. This works on the person after they've been born. So, for example, your muscle cell, your liver cell. Somatic editing is the kind that we use in treating sickle cell anemia, HIV, cystic fibrosis, and it's the kind of edit where the person they can they can agree to their genes being edited, they know what is going on, and it will not affect the person's future generations like germline editing can. So currently we use CRISPR in medicine, agriculture, livestock, and genome research. In terms of medicine, we're using it of course to treat things like sickle cell anemia, HIV, even certain cancers is being used. CRISPR is being used to treat it. And in terms of agriculture, we can use it to make our crops bigger, maybe bigger fruits. We can also use it to make the crops less bitter. So mustard greens has a lot of nutritional benefits, and recently scientists used CRISPR to make it taste less bitter because it tastes so bad and people don't want to eat it. But they have been able to cut out the gene that makes mustard green taste bitter, and people are now able to eat it, put it in their salad, and it doesn't taste as bad, and also they can get the nutritional benefits that it gives. In terms of livestock, CRISPR can help animals like chickens to be resistant to the avian flu. We can also use it to improve maybe meat quality, the wool quality of animals like sheep's. And in terms of genome research, we can turn genes on and off and see how we can turn off certain diseases in a person, certain genetic diseases in a person. In terms of future uses, we can only speculate what can we use CRISPR for in the future. However, we can speculate that we can use it to eliminate genetic disorders entirely. We can use it to maybe engineer mosquitoes that can't transmit malaria, because malaria is still one of the like one of the easiest things we can not eradicate, but one of the easiest things we can sort of cut out if possible. We can also maybe use it to enhance human traits like making a person taller, making a person's eye color a different color. We can maybe use it to make a person more intelligent. And we can also use CRISPR to tailor gene therapies to a specific person based on their DNA. And even maybe reprogram certain cancers to be less drug resistant. So the three people that are mostly associated with like the origins of CRISPR are this guy on the right, Francisco Mujica, and these two ladies, Jennifer and Emmanuel. Francisco was the first guy. He was a microbiologist in the University of Alicante, and he was credited with discovering CRISPR in bacteria. So he saw that bacteria had repeating sequences. And the two ladies down here, Jennifer and Emmanuel, they developed CRISPR-Cas9, the system used for genome editing. And they were the ones to realize that Cas9, that enzyme, can be used to cut the DNA at a certain place, at a certain sequence to edit the gene. In 2020, Jennifer and Emmanuel, they won a Nobel Prize in chemistry. So we can say that CRISPR is advantageous, but also there can be disadvantages because CRISPR is so powerful. It raises some concerns, but on the positive side, we can say it would eliminate inherited diseases. It can correct faulty genes. Maybe we can even improve food security by always maybe having the best crop yield. We can help use it in scientific research. It can help scientists understand how genes function. We can make medicine more precise. But also the slippery slope for CRISPR is when we start to get too off-target edits. We don't know what CRISPR might accidentally cut out because we haven't used CRISPR for such a long time. It's not foolproof. And also, should we be editing certain genes? Should we be editing embryos before they're born? Because those kind of edits can affect a person's gene pool permanently. They can affect a person's future offsprings. Also, we can think about the fact that CRISPR might create an inequality in healthcare access because a rich family might be able to get their child's cystic fibrosis cured while a poor family might not. And also, there's this slippery slope of designer babies. Once parents figure out they can edit their kids' genes, what if they say, oh, I want my kids to be taller? I want my kids to be smarter. I want them to have green eyes instead of brown eyes. I want them to have straight hair. So those are some of the advantages and disadvantages to think about in terms of CRISPR. It is very powerful. It is a breakthrough in science, but also it's important to sort of consider the benefits and the risks of CRISPR. So in 2018, there was a controversy in regards to gene editing. A Chinese scientist, his name is He Jiankui. He was in a major controversy because he edited the genes of human embryos. So these kids weren't even born yet. The name of the kids were Lulu and Nana. And he made them resistant to HIV. So he edited their genes to be resistant to HIV. And while he did all of this, he made so many mistakes, broke so many rules. He bypassed ethical regulations, he forged documents, and tricked doctors into implanting embryos that were genetically edited. The parents didn't even know. And years later, people are coming to conclusions that what if his edits actually did more harm than good? What if those kids, they didn't need those edits? The parents weren't aware. The kids couldn't even, the embryos couldn't consent to the edits being made. And what if his edits have unintended consequences? So his actions sparked outrage in China, and he was sent to prison for 3 years. And with his controversy, it sort of makes the science community ask the question: should we edit embryos? What counts as necessary? What is necessary to edit? And who decides what is ethical and what isn't? And who also gets to edit the genes of maybe kids, maybe adults, maybe embryos? So these are things that we need to keep in mind. So, just to conclude, I want to leave this here. It says, with great power comes great responsibility. Who decides what is ethical and who decides what we can and what we should edit? And also, think about it: who is consenting? What if the kids don't consent when they grow up? What if the kids didn't even want this to be done to them? So, these are just certain things to keep in mind. I hope this was informative, and thank you for listening.